one of the questions I ask myself very often is what are some of the changes in medicine that can change medicine for everyone all over the world? The reason I ask this question very often is that we've all lost people to disease. So in Africa, disease has had a very huge impact in our lives. I've lost so many people in my life, and I know all of you have lost people as well. And all over the world, people are curious. People are curious about who we are as humans. People are curious about what is disease. And so, I just want to ask by looking at the past to make us think of what are those big changes in medicine that have ever happened. And I know you can imagine so many different kinds of things that have happened. But for me, I think one of the most transformational moments in medicine was the realization that disease are caused by germs or have a cause, that disease is not magic. And this is actually an observation that was first made by Imhotep, who was regarded as a god of medicine in Egypt. I think the other transformational event in medicine was the discovery of vaccines. And you can see from the moment that humans realized that diseases have a cause to the moment that vaccines were discovered was centuries, like hundreds of years passed to that moment. And the other, I think, also was the discovery of microbes. So Louis Pasteur, being one of the major founders of the field of bacteriology. But I think in spite of the many revolutions that have occurred in medicine, the fact is that most diseases, in fact, have no cure. But even sadly, for those few diseases where there's a cure, not all of the medicines work in all people. And while these statistics here may not be so accurate, because we haven't done research to know how effective medicines are across a different range of diseases. But the fact is that the state of medicine is very bad, because even the medicines that we have don't work in all people. So I always thought that we need to think of ways to change how we discover medicines. And I think we need to change who discovers medicine, from the traditional scientists in a white lab coat. How can we engage people in various areas to be part of the process of medical discovery? And we also need to change how we discover medicines. And also where medical discovery occurs. And I think discovery needs to engage a wider array of people in a cycle. So from researchers who mostly think about what's next, to students who are learning, are in the process of learning about the future. And maybe to give an example, I want to go back to 2003, which is about 12 years ago, when I was a student at a university in Kenya. And 2003 was very different from where we are now, because in 2003, only a few people in the world had mobile phones. In fact, within my university, there was no single student with a mobile phone in 2003. Computers were just beginning to emerge in Kenya, so students again didn't own computers in 2003. But I was working with a scientist at a private research facility who was studying these viruses, virus-like DNA, that occur in all our genetic material. So they're called endogenous retroviruses. They constitute about 10% of the genome. And so when I was working with this particular scientist, I was looking at how these retrovirus-like material might be important in pregnancy, I became immediately interested in how these viruses might interact with HIV, which is a retrovirus as well. And so I asked the scientist, that, is there a way I can test this idea? And the scientist said, this is a very interesting idea, but it's going to cost a lot of money. We are going to need a lab. So I don't think we can do it. But I was very interested in this because I had a hypothesis. The hypothesis was very simple. That if there's, in our DNA, material that looks like retroviruses, is it possible that these retroviruses could recombine with HIV? Could they combine with HIV to form new viruses that might then evade antiretroviral drugs, for instance? So it came upon me that maybe if I had a way of testing this idea on a computer, then maybe it could be a way in which I could get an answer quickly. So I had no computer, but fortunately we had cyber cafe. We had one cyber cafe on campus. So you could go to the cyber cafe and for about $1, you'd be able to use the computer for a period of 1 hour. Now $1 in 2003 was much more money than I would spend on food in 3 days. But I really wanted to test this question, so I went to the cyber cafe each day, and thankfully people at the cyber cafe were very friendly. So they would let me browse for free after I paid for a session, and then I browsed for extra hours for free. But anyway, through going to the cyber cafe, in about 6 months, I learned how to analyze DNA because there was already human DNA sequences on the web. So I could go and look at human DNA sequences and try to look at those retrovirus-like regions in the DNA. And so from this, I was able to test my idea, which was basically showed that indeed the retrovirus-like sequences in our own genomes, if they could recombine with HIV, they could potentially recreate new viral material that could evade antiretroviral drugs. So I've always been interested, how can we combine learning with discovery? So I continued pursuing this idea, so here with a team called Fit to Cure. And the idea with Fit to Cure was very simple. The idea was to develop a game which abstracts some of the steps in the drug discovery process in a way that students can learn some elements of how drugs work, but at the same time potentially contribute new knowledge to understand how drugs interact with proteins at a molecular level. So we developed this game which we tested with a number of students here at Notre Dame. And the idea was that as students play the game, we collect data about how students fit the protein, the drug molecules into the protein. And so from that data we can build better algorithms that can identify what drugs will be more potent in blocking different kind of proteins. So I think because there's so many diseases in the world, I think if we can find ways in which we can engage a larger number of people in the discovery process, it could help us address the diseases that affect even the poorest regions of the world. So to test this idea, last year we organized a workshop, so together with some friends. So here, Frederick, Sheila, Simon, and we basically asked the idea if you could bring about 20 students into a room and develop kind of a cloud-based platform where they can all have access to data, so in this case, data from Ebola virus, and try to test a hypothesis of how, by analyzing Ebola virus sequences, you can be able to understand some specific knowledge about the virus. And here is just to show so the kind of platform that we were envisioning. And the idea was that we'd have a cloud computing platform that would host like data from Ebola virus, and then integrate that with some software tools. So basic software like R for statistical analysis, and allow students to be able to interact with this data. So basically write code, be able to analyze this data, and also derive some insights. And the specific hypothesis I wanted to test was actually, given that we have a very large number of DNA sequences of pathogens on the web, is there a way we can use the DNA sequences of pathogens to predict what human populations might be more susceptible to a given viral infection? So because response to many viruses is mediated by a huge class of genes called the HLA, which are very important in how we respond to viruses. And depending on which HLA alleles you have, you might be either susceptible or more resistant to a viral infection. And so basically the way this approach was going to work is that basically we have a computational method that can take a viral sequence, and from that viral sequence we can make predictions of what HLA, so this HLA is a very large group of genes that determine how we respond to viruses, but we would be able to predict based on the viral or protein sequence whether a given HLA allele would mediate a protective or susceptible response to the virus. And so, to test this idea within a virtual setup, we basically first began by training the students on Ebola biology. So the idea behind this was that we take a real world problem, but at the same time we teach students how to tackle the problem. From basically learning about the biology of the Ebola virus, learning about the genomics of the virus, and also learning about how to write basic code and how to do simple statistics. And in this process, our goal was how can we combine the process of learning with the research and discovery. But I think perhaps most importantly, I wonder whether we need to shift the way we think about disease, because the germ theory, I think, took us to thinking about disease as something that is almost external to humans themselves. So when we study disease, we tend to study the germ more than the human. So if you look at most drugs, for instance, they developed the context of killing the disease-causing organism, but with very little attention to the human role in the disease. So I think we need to think of disease more widely. I hope this is something that's going to be more possible as we study more human populations. To look at a disease from a wider perspective by looking at the role of genes in disease, for instance. And so I think even as we talk about precision medicine and other kind of medical revolutions, it is my hope that we are going to be able to engage people all over the world, especially in Africa. So this is why I've moved back to Africa, to see how we can engage other students to be part of this process. Thank you.